Hello and welcome to GED Live at Pals, to another GED Science screencast. Today we will have a look at a few important concepts in science that you really should know about. This is like a short summary in this screencast of important topics. If you want to know more of some of the topics discussed here, there are individual screencasts on each of these topics in our library. So if you want to know more about a topic, just check out our library. All right, let's get started. Okay, so what we will talk about first of all in this short screencast here is we will have a look at the DNA structure and composition, what DNA is and what it's made of, and what its structure is, and a little bit about its function as well. We will have a look at animal and plant cells and their structures and their similarities and differences. And we will look at two different types of cell division, mitosis and meiosis, what their similarities and differences are. And we will look at photosynthesis and cellular respiration. Okay, so first bit, DNA structure and composition. So DNA, first of all, is a nucleic acid. Nucleic acids are one of the important biological macromolecules besides carbohydrates, lipids, and proteins. So DNA is a nucleic acid. What does DNA stand for? So DNA is a specific type of nucleic acid. It is a deoxyribonucleic acid. And it contains the instructions an organism needs to develop, live, and reproduce. DNA is found in almost every cell of an organism and is passed on from generation to generation. Almost every cell of an organism, if we think about our own body, red blood cells, for example, do not contain DNA because they are highly specialized to transport oxygen, and all the space available in a red blood cell is filled with hemoglobin to transport the oxygen around. So oxygen, red blood cells, they don't have a DNA. And DNA is found inside the nucleus of a cell. So red blood cells don't have a nucleus either. Most of our other body cells, they do have a nucleus and they do have DNA. Okay, so a little bit on the structure of DNA. We can see how DNA is organized here. We can see a chromosome, and a chromosome is basically in this shape a very, very condensed form of DNA. It's a very long molecule coiled up into this chromosome shape. And we can see that the DNA has this typical alpha helix structure, this helical shape. We have the notes here on the side. DNA is a double strand twisted into a helical shape. DNA is organized in chromosomes. A chromosome is a large molecule of DNA with associated proteins around which the DNA is wrapped. Chromosomes in this X-like shape you've probably seen before. These chromosomes only exist in this form during cell division, which we will talk about afterwards. So when a cell divides, these two halves will split and they will go to opposite ends. So it's very important that the chromosome shape is like this during cell division, because DNA is moved around in a cell, so it needs to be packed up. So a chromosome is basically a very, very packed up form of DNA ready to be moved around the cell. Just to give you some kind of context here, why is it important to pack the DNA up before it's moved around the cell? A single cell of your body has about 2 meters of DNA in it. A total of 46 chromosomes in one single cell, 2 meters of DNA. So it's very long and very, very thin, so DNA is quite fragile as well. And for sure, before you move these 2 meters of mass through the cell, I like to compare to a plate of spaghetti, well, you have to wrap it up first and pack it up so that you can move it around. So you have to pack it up first so that you can move it around the cell. So let's have a closer look at the DNA. The DNA building blocks, what DNA is made of, are so-called nucleotides. Nucleotides have three main parts. We can see nucleotide pairs down here. Each nucleotide has a phosphate group and a sugar. The sugar is the deoxyribose, and one of four nucleobases. So what all four different nucleotides have in common is the phosphate and the sugar. The phosphate and the sugar form this backbone of a DNA strand. So these two DNA strands, the backbone of it is formed by the sugar and the phosphates. And inside the middle, the bases meet. We can see up here that always A pairs with T and G always pairs with C. This is so-called complementary base pairing. And yeah, adenine always pairs with thymine and guanine always pairs with cytosine. The type of bonds that hold the two DNA strands together are weak bonds, so-called hydrogen bonds. They're not fully strong chemical bonds like covalent bonds or ionic bonds. They are a type of a weak chemical bond, so-called weak force. In this case, hydrogen bonds. Hydrogen bonds are some of the stronger weak forces. So our DNA double strand is held together by hydrogen bonds, which form between the complementary bases. We can see as well that we have two hydrogen bonds between adenine and thymine forming, and three hydrogen bonds between guanine and cytosine. And yeah, one more definition. What is a gene? A gene is a specific length of DNA which codes for a protein. And proteins then in return produce the traits that our body, we as an organism, have. So a specific sequence of nucleobases in our DNA will in the end does contain the information to make a protein. The cell can extract that information and then use the information to build that protein. The protein itself is built at ribosomes outside of the nucleus. And that brings us to cell structures. We'll have a look at animal and plant cells. Here we have a very detailed diagram of a plant cell on the left-hand side and an animal cell on the right-hand side. We can have a look what these two cells have in common. So first of all, both cells have a nucleus. Again, both cells have DNA. Very important. It contains all the information for the cell to grow, to survive, to live, to reproduce. So both cells need that. The nucleus itself is encapsulated by its own membrane. This membrane is called the nuclear envelope, and it has pores so that substances can easily exchange between the outside and the inside of the nucleus. Inside the nucleus we have a region which is called the nucleolus, which is a bit darker region if viewed under a microscope, and if the nucleus is stained, the nucleolus is a darker region inside the nucleus. We have inside the nucleus the chromatin, which are elongated chromosomes, so it's basically DNA. Then we have mitochondria. Mitochondria is the plural, many mitochondria, one mitochondrion. Mitochondria are important for the cell because this is where cellular respiration happens, about which we will talk at the end. We can see vacuoles in animal cells and in plant cells. We have a quite big difference here. Although both cells have vacuoles, plant cells have a very large central vacuole, which is as well a permanent vacuole. It's always there in plant cells, whereas in animal cells, these vacuoles form temporarily and can then vanish again. Ribosomes are found in both animal and plant cells, and ribosomes are the structures that construct the proteins. They're able to decipher the code of DNA and use that information to build the protein DNA is coding for. Lysosomes are only found in animal cells, and they contain usually digestive enzymes to break things down. We have a Golgi apparatus. Golgi apparatus function is to pack up substances inside the cell, usually destined to leave the cell. Like, for example, a lysosome could be built by a Golgi apparatus. The lysosome travels through the cell, eventually fuses with the cell membrane, and the contents of the lysosomes, the digestive enzymes, are given to the outside. So the Golgi apparatus very often produces secretory vesicles. Then we have smooth endoplasmic reticulum and the rough endoplasmic reticulum. Rough endoplasmic reticulum is usually found very close to the nucleus, and it contains many ribosomes, so it's important in the making of proteins and in the packaging and modifying of proteins. We have the cytoplasm. I did not mention that. The cytoplasm is the stuff that fills the cell. It's like a jelly-like liquid, which, inside which many of the cell's metabolic reactions occur. And what else do we have for sure? We have the plasma membrane or the cell membrane. It says down here it's the same thing. Cell membrane, plasma membrane. It's a term we can use interchangeably. They are the same thing. This plasma membrane, the cell membrane, separates the inside of the cell from the outside, and it has a very important function in controlling what things are able to get into the cell or out of the cell. It allows the cell to create its own internal environment, which is different from its surrounding. So let's have a look at some structures that are specific to plant cells. We already said we have the central vacuole, which is not labeled as green here. It should probably, because this kind of vacuole is really just in plant cells. Again, animal cells have smaller vacuoles which are not permanent. Plant cells have a cell wall. The cell wall is a very rigid outer structure that gives a plant cell its fixed shape and rigidity and strength. The cell wall usually works together with a cell vacuole. There's a large vacuole inside the cell to give the plant cell stability. The vacuole usually draws in water and really pushes against everything inside the cell, which makes it a little bit blown up, and it pushes against its own cell wall from the inside, making plant cells always a little bit blown up and really rigid and strong. And then we have chloroplasts. Chloroplasts you have probably heard about. Chloroplasts are the structures that allow plant cells to do photosynthesis. Photosynthesis, the process by which plants make their own food in the form of glucose. We will talk about that later. In animal cells, we have a structure which are the centrioles. Centrioles are important when it comes to cell division, mitosis, which is a little bit different in animal and plant cells. Okay, so one more time, let's have a look at a few of the very important structures that you definitely should remember about. The cell membrane, it controls the movement of substances into and out of the cell. It's in plant and animal cells. The cytoplasm, jelly-like substance where chemical reactions happen in plant and animal cells. The nucleus, which carries the genetic information, the DNA, and controls what happens inside cells, again in plant and animal cells. We have the mitochondria, where cellular respiration is happening, or most of the cellular respiration, where energy is released from food. We have the central vacuole in plant cells, which contains a liquid called the cell sap, which keeps the cell firm by pushing against the cell wall from the inside. In plant cells only, the large central vacuole, again, other vacuoles are found in animal cells as well. And we have the cell wall, which is made of a tough substance called cellulose and which supports the cell and gives it its rigidity. Okay, cell division, mitosis and meiosis. First of all, cell division is the process by which a cell or a parent cell divides usually into two daughter cells. These two daughter cells form in mitosis and meiosis as well. Meiosis, we have two divisions leading to four daughter cells, but step by step. So mitosis is a type of cell division that results in two daughter cells, each having the same number and same kind of chromosomes as the parent nucleus. Mitosis usually is for growth in an organism when the organism increases its number of cells, for repair of damaged tissue, or replacement of cells that have died. Meiosis is a type of cell division that results in four daughter cells, each with half of the number of chromosomes of the parent cell. And meiosis is happening for the production of gametes. Gametes are sex cells, sperm, and egg cells. So meiosis is a very, very specific process for sexual reproduction to produce the reproductive cells, the egg cells and the sperm cells. Whereas mitosis is happening in our body all the time as well, again for growth and repair and replacement of worn-out cells. So again, there's a lot more to say. If you want to know more about mitosis and meiosis, again, check out our other screencasts. Here, very summarized quickly, we have a parent cell. Before the parent cell can divide, it needs to duplicate its DNA so that both daughter cells can have the same amount of DNA as the parent cell had before. So if we look at the parent cell, we see we have 1, 2, 3, 4 chromosomes, and we can see the daughter cells have these 1, 2, 3, 4 chromosomes as well. So that this is possible, chromosomes need to replicate, or the DNA needs to replicate. So that happens. The replicated bits of DNA, they stick together, and we get our typically X-shaped chromosomes, which we saw earlier as well in this diagram here. So here we have already a replicated chromosome. One side is one copy and the other side is the replicated copy of DNA. We can see these X-like structures here, a duplicated chromosome with two sister chromatids. During mitosis, we have a couple of phases. We have prophase, where these chromosomes become visible. The chromatin, the elongated DNA, condenses to form the chromosomes. Then we have the second phase of mitosis, the metaphase. Here our chromosomes get lined up in the center or on the equator of the cell by the centrioles, which form the mitotic spindle, and we have we can see these microtubule fibers that move the chromosomes around and line them up on the equator. And now during anaphase, that's actually not shown here. During anaphase, the chromosomes are pulled towards the poles. They're split in the middle, the sister chromatids are split, and the halves are pulled towards the poles of the cell. One half is pulled to the left side, the other half is pulled towards the right side. And then during telophase, a new nucleus forms, and then the cell divides in a process called cytokinesis, and we get two daughter cells that have the same genetic material as the parent cell. Now, meiosis is a bit more complex. During meiosis, again, we need, first of all, the parent cell to duplicate its chromosomes, its DNA. What happens then is that the homologous chromosomes, the chromosome pairs, they will form so-called tetrates, where they stick together. During that process, or during this formation of the tetrate, the homologous chromosomes may exchange genetic material, which is a process that is called crossing over. The tetrates then line up on the equator, similar to mitosis, only that we can see now the homologous chromosomes are still sticking together. We can see some of them are overlapped here, so this is the crossing over process where pieces of them are exchanged. And now the whole chromosome, the two chromatids still sticking together, they are pulled towards the poles of the cell. Very important here that the way the chromosomes or the tetrates line up is random in terms of how they are orientated. So one of them could be flipped as well, having the blue side on the other side. This is called random assortment, and this is one of the very big sources of variation in the gametes in the end, which makes the gametes genetically different every time this process happens. Once the homologous chromosomes got separated, we get two daughter cells, and these two daughter cells then undergo a second step of meiosis, which is called meiosis 2. And there, similar as in mitosis, the halves of the chromosomes get separated, and we in the end get 4 cells which are genetically different from each other. And they will be genetically different from other gametes that have been produced before. Again, why is that? Due to the random assortment of the chromosome tetrates in this step. Again, the way whether the blue or the red chromosome, the mother's or the father's chromosome is on the left or the right side is completely random. We humans have 23 different types of chromosomes, so 46 in total. So there are 23 of these pairs lined up here. So there is a total of 2 to the power of 23 varieties or possible combinations here, not including the actual genetic exchange in crossing over. So they are basically unlimited possibilities. So one more contrasting of mitosis and meiosis. DNA replication happens in both in mitosis and meiosis before it begins. In mitosis we have 1 cell division, in meiosis we have 2 cell divisions, meiosis 1 and meiosis 2. The tetrates or the synapses of homologous chromosomes only occurs in meiosis, along with the crossing over between the non-sister chromatids between the homologous chromosomes in prophase 1. The number of daughter cells and genetic composition. In mitosis, we have 2 diploid daughter cells, which means they have the full set of chromosomes. We can see they're diploid 2n because we have 2 of each chromosome, 1 chromosome from the father, 1 from the mother. That's what we call a diploid. And we can see in meiosis we have 4 daughter cells which are haploid. So they each containing half as many chromosomes as the parent cell. Daughter cells are genetically different from the parent cell and genetically different from each other. The role in the animal body. Mitosis produces cells for growth and repair and replacement, whereas meiosis produces gametes, the sex cells, sperm and egg cells, and assures genetic diversity due to sexual reproduction. Photosynthesis, a cellular process, and respiration, specifically cellular respiration and specifically aerobic respiration. We will have a look at photosynthesis. Let's have a look at this first, or at the same time actually. Photosynthesis is a cellular process in plants which converts carbon dioxide and water into glucose and oxygen, making use of the energy from sunlight to make it possible, to make it happen. this reaction. So two simple molecules, carbon dioxide and water, are turned into a complex molecule, glucose, which is food, which is fuel that contains a lot of chemical energy. To get that energy into the glucose, we need an energy source, which in this case is the energy of the sun. Oxygen is a byproduct made in this reaction. Cellular respiration, on the other hand, is a process, or is the process which breaks the glucose down with the help of oxygen to release energy for cellular processes. Carbon dioxide and water are the products of this reaction. Photosynthesis happens only in plant cells, and it happens inside the chloroplasts of the plant cell, and it is a so-called endothermic reaction because it absorbs energy from the surroundings, specifically absorbs light energy. We see the equation one more time. Carbon dioxide and water react to form glucose and oxygen. Whereas in aerobic respiration, this happens inside plant and animal cells. Both cells need to be able to break down their food, their glucose, and it happens inside the mitochondria, at least aerobic respiration. Aerobic respiration means it makes use of oxygen to break down the glucose, and this happens in the mitochondria of a cell. It's an exothermic reaction because it releases energy to the surrounding. In this case, the oxygen is broken down with the help of glucose. Glucose is broken down with the help of oxygen. Carbon dioxide and water are produced, and the energy from the glucose is released, and the energy is immediately used to make a substance called ATP. ATP is basically the energy currency of a cell. The cell uses ATP inside the cell whenever it needs energy. For any process that needs energy, the cell has ATP available, and that ATP is supplied by cellular respiration. So it is really important to understand that photosynthesis and cellular respiration are very closely related to each other. If you know one of the processes, you do know the other process as well. Look at the equations. They are the opposite. The reactants of photosynthesis, carbon dioxide and water, are the products of respiration, carbon dioxide and water, whereas the products of photosynthesis, glucose and oxygen, are the reactants of aerobic respiration. The big difference is the way that energy flows here, and the way that energy is utilized. In photosynthesis, the energy comes from light energy, and that energy is in a quite complex way transferred into the glucose and is now stored as chemical energy. Chemical energy is a energy storage, and that chemical energy is then released, made available to the cell in cellular respiration in the form of ATP. All right, that's it from me. 4 concepts in life science that are very important for the GED test. Again, if you want to know more about the topics, check out our library. Every topic has its own dedicated screencast where I go into much more detail. If you found this video helpful, please consider subscribing to our YouTube channel. It allows us to reach a lot more people like you who want to prepare for their GED tests. I wish you all the best for the future, and I hope you pass your GED science. And I will see you in part 2 of important concepts in science that you need to know to pass the GED. Have a good day.